Okay, everyone, welcome back to session 10, Basic Science Integrated into Therapeutic Management. I'm very happy that we have a really nice row of speakers, and we have various topics covered, from how kidney cancers are immunogenic to predicting therapy and what to consider when designing biomarkers. So let's start with the first presentation. I welcome Florent Petit-Pré, who will speak on TLS and B-cell responses in RCC.
Hello, everyone. So thank you to the organizer for giving me the opportunity to present to you today. I will talk today about B cells and tertiary infrastructures in kidney cancer. So this is my outline. I will first briefly introduce B cells and tertiary infrastructures in the tumor microenvironment in general, and then we'll go back to a study in particular in tertiary infrastructure in kidney cancer. So the tumor microenvironment has to be considered as a whole system. In a tumor, we don't just have the tumor cells themselves, but we have virtually any kind of immune cell that can be present. We also have the fibrous and endothelial cells forming blood vessels. It's really important to study the composition of this tumor microenvironment because it has been shown to be associated with the clinical outcome in many different cancer types. Inside this tumor microenvironment, we can sometimes find structures called tertiary infrastructures. These TLS, I will just call them TLS for short in the rest of my talk, are lymph node-like aggregates that are composed of a B cell zone that in mature TLS is actually a functioning germinal center. We have a T cell zone, and we have nearly all follicular population that can be present, like follicular helper T cells or follicular dendritic cells. And interestingly, the presence of this tertiary infrastructure has been shown to be associated with response to immunotherapy in different cancer types, and we notably showed it in soft tissue sarcoma. So these are results from a phase 2 clinical trial called IKCSEU23 that was run at the MD Anderson Cancer Center. And those were advanced soft tissue sarcoma patients treated with pembrolizumab. And as we see, patients that were associated with what we call sarcoma immune classes E and D, so those that were enriched in immune cells and that notably had the presence of tertiary infrastructures were associated with a better response to the therapy and a better progression-free survival under treatment. And interestingly, this result is not only restricted to soft tissue sarcoma, and it has since been shown in many different cancer types, such as sarcoma, melanoma, clear stain and renal cell carcinoma, lung cancer, and also in a pan-cancer cohort, where the presence of mature tertiary infrastructures was associated with a better response to immune checkpoint inhibitors. But now let's go back into detail in the kidney cancer case. So this comes from a study, so the first author of that study was Maxime Mélan, a rising star postdoc who was doing that work in Paris and is now in Boston. And so we used spatial transcriptomics to study the tumor microenvironment of kidney cancer. And we first separated the tumors based on their TLS status, so those that had TLS and the tumors that had no TLS. And interestingly, in the tumors that had some tertiary infrastructures, we could see a very strongly organized tumor microenvironment. So notably for B cells and T cells, but also for fibroblasts, we saw that we had a strongly organized tumor microenvironment. So on the left here, I'm not sure I have a pointer, so no problem. But on the left, the yellow dotted zone is actually where the TLS are located, as confirmed by a pathologist. And then we looked in particular at what kind of B-cell subsets we could see inside these tertiary infrastructures, these TLS-positive tumors. And we saw that naive B cells were not particularly strongly associated with a particular location inside the tumor. So just to make sure here on this slide, the left part is the expression of a TLS-specific gene signature. So the red areas are actually where the TLS are located in the tumor. And when we go further down the maturation stages of the B cells, we see that we have some memory B cells that are located inside the tertiary infrastructures. The germinal center B cells are all, as could be expected, inside the tertiary infrastructures only. And we see some plasmablasts really restricted to the TLS, but when we go to full-blown plasma cells, they are not only found inside the TLS, but are also spread out across the tumor. We looked also at the different classes of immunoglobulins that could be expressed by these potential B cells and plasma cells. And so there is a problem with the image. No, so you will have to believe my word for it. But actually, we found that inside tertiary infrastructures, we had class switch happening. So maturation of the B cells inside the tertiary infrastructures, and that only mature and class switch B cells were distributed through the tumor. So I encourage you now to go and see the paper if you want to see those images. And then we looked at how these plasma cells and class switch plasma cells could be distributed throughout the tumor. And we found that plasma cells, and mostly IgG positive plasma cells, were actually colocalizing with CXCL12 positive fibroblasts. So actually, from the TLS and throughout the tumor, we have tracks of fibroblasts that will lead plasma cells through the tumor. Using spatial transcriptomics, we could also look at the B cell repertoire of these tumors, and also in a spatially resolved way. So we are able to see that inside the tertiary infrastructures, we had a higher clone type counts, and we had a higher mutation counts in the rest of the tumor than actually just in the tertiary infrastructures. So all of this indicates that inside the tertiary infrastructures, we could have somatic hypermutation. So selection of clones specific for some antigens inside the tertiary infrastructures, and then that they would be disseminated in the tumor. And actually, on the right of the slide, you have, so these are the same clone family that are represented. And we didn't expect to be able to see it, but we were lucky enough to see that. So each of those images is one different clone family, and we see that we have those two clone families that are highly represented where the tertiary infrastructures are. But we also saw clones from the same family. So very, very close B cell receptor. So coming from the same clone, just with a different mutation. So we really see that we have this dispersion of clones selected inside the tertiary infrastructures throughout the whole tumor. And why is this interesting? Because then we looked at the tumor cells, and we saw that in TLS positive tumors, a lot of these tumor cells were actually marked by IgG antibodies. So we would have secretion of anti-tumor antibodies inside the tumor. And next to these antibody-bound tumor cells, we saw a lot of those antibody-bound tumor cells were undergoing apoptosis. And we looked at what kind of cells could cause this apoptosis, and we found a strong presence of C68 positive macrophages next to these apoptotic tumor cells. So basically, we have those plasma cells generating antibodies against tumor cells, and then those tumor cells are marked for apoptosis by LDCC through macrophages. And now, what's the more profound clinical message that we can have from these plasma cells inside the tumors? So we've looked at data from the Bionic clinical trial. Jan, I will talk to you a bit more about this trial later. And we saw that actually in the tumor that had a stronger IgG staining of the tumor cells, we also had a much stronger response rate to nivolumab and ipilimumab, or nivolumab alone. So we have a stronger response rate to immune checkpoint inhibitors in those tumors that have a stronger presence of plasma cells. Interestingly, this is not, again, restricted to kidney cancer, but since similar results have been shown in lung cancer and soft tissue sarcoma. So briefly, the take-home message is that in some tumors, we can find tertiary infrastructures with lymph node-like aggregates forming in the tumors, and that those are sites of activation or maturation of a potent immune response against the cancer. And they are associated with an increased survival and a response to immune checkpoint inhibitors in many different cancer types, including clear cell renal cell carcinoma. And in clear cell renal cell carcinoma, we showed that these tertiary infrastructures organize their tumor microenvironment and that they generate and propagate anti-tumor plasma cells that will lead to a higher apoptosis of tumor cells and a higher response to immune checkpoint inhibitors. So with that, I'd like to thank all these amazing scientists I had the chance to work with, and all of you for your attention.
Thank you. As our second speaker, I would like to welcome Hayden Kissick from Emory University, talking about circulating T-cell activation as predictor of therapy response.
Great. Thanks so much for having me. I'm going to talk a lot about what's happening inside the tumor as well, but what the main take-home message here is that very quickly in the blood after immunotherapy, you can learn a lot about if a patient is going to respond or not. This is a small group of patients we were just studying, and I think you've probably all seen a cohort like this before, 40 patients, 10% complete response, 20% have some sort of partial response, half of them maybe no response at all. And it's a kind of ongoing question, which patients need to stay on drug, which patients need to move on, which ones should maybe have never got this in the first place. We're basic immunologists. Our lab understands very fundamental things about how the immune system works, what sort of things happen. We were just studying activated T cells in the blood of these patients. What we have here is just gated down to the CD8 T cells in the blood of a patient. We're looking at two markers, CD38 and HLA-DR. These tell you if a T cell was recently activated. So if you look in all of your blood right now, you've got a few of these cells activating, responding to subclinical sorts of things. When you have a vaccine or something like that, you have a big burst of these cells in the blood that are responding, becoming effectors, turning into memory cells and things like that. What we see when a patient gets anti-PD-1 therapy or any of the sort of IO combinations, there's a big burst of activation of these activated cells in their blood extremely quickly after the therapy. So what I'm showing here is 2 cycles. When they come in for their second infusion, we're taking blood just before it, we see this massive sort of burst of activated cells in their blood. And if I show you the summary data for all our patients, pretty much everyone has some level of activation. Some have small, some have large. It's a big mix. It's always the first cycle though. We don't often see big, if they didn't sort of activate on the first, we don't see many on the second, third, fourth, and it's very rare that it goes up again. It's usually biggest burst early and then sort of lower moving on. What's important though is the size of this burst on the first cycle of treatment is very good at predicting who will have a response to therapy. So this is just a waterfall plot. Orange is an optimal cut. Above that cut, they had more activation. Below that cut, they had less. You can see very strong, clearly, all our patients with the strong tumor regression had this activation in their blood on the first cycle. I can show you spider plots. This is sort of, plays out with many patients. And then there's an overall survival benefit, progression-free survival benefit, where we can actually sort of predict on the first cycle who is going to have a clinical benefit from these. I will point out one interesting thing. Most of these bloods are taken at 3 or 4 weeks, depending on the cycle. If we take blood at 1 week after, we're much more able to find activation and measure this more accurately. Immune system responds on very short half-lives, days, maybe a week. So this sort of 3-4 weeks is challenging for us. We do think you'll notice those patients, the 2 who had the complete response and no activation, we do sort of see other activating markers in their blood, and we think maybe we missed the window on them. So getting blood early is really important. Sort of an interesting underlying biological question is, why do some patients have an immunological response or not? This is work from our lab, basic immunology. I'm going to take you through just the most important part of this. What we've found is T cells undergo activation in many different stages. There's this important type of T cell we call a stem-like T cell that's kind of the source of all the effector anti-cancer cells. This cell resides in the tumour, and this is the cell that PD-1 blockade works on. It works on this cell to make effector progeny cells. We can find these cells in the tumour very easily. We can look at almost any cancer. We've done kidney, liver, brain, lung, melanoma. Almost every cancer, these cells exist, a stem and an effector cell. If you sort them out, they have very different functions. The effector cell is cytotoxic, has granzyme B, all the things you think of for killing. The stem cell has none of those, but it has very good proliferative capacity. And so when we stimulate that stem, it turns into the cytotoxic daughter cell, and then that is the one that does the killing. And as I say, that's how PD-1 sort of works. And so it's important to have these cells in your tumour. They don't just randomly float around in the tumour. They reside in a very defined location, which we call an antigen-presenting niche. It's a little bit like the TLSs you just heard about, but different in its own way. In this niche here, I think this is actually a prostate cancer patient, but every tumour that we look at has these niches if you have an immune response to your cancer. You have stem CD8 cells, stem CD4 cells, the green cells are antigen-presenting cells, and as I say, this is across pretty much every single cancer type. What's interesting though is those patients that have an immunological and clinical response to their tumour, sometimes years after the tumour was taken out, have strong evidence of this happening in their tumour at the time of surgery. So this is two patients, one came in with sort of advanced disease, obviously probably going to go into immunotherapy at some point, had a complete response after a couple of cycles. The patient at the bottom, same sort of situation, came in, absolutely no response whatsoever. If we quantitatively image these different types of cells in their tumour, this responding patient, long before they received PD-1, their cancer had an immunogenic phenotype. So these plots are showing in red where the T cells are in that patient's tumour, blue is these stem cells, and then the orange is sort of where these little immune niches are where the cells are residing. So you can see this top patient, very immunogenic tumour, bottom patient, at the time of surgery, really nothing at all. And then when we look in their blood, 6 months or longer later, the ones that had an immunogenic tumour at the time of surgery also had an immune response to their cancer. So we see this burst of activation in that patient's blood, whereas the other patient really had nothing at all. This plays out in a relatively small group of patients, but if you have more of these niches and stem cells, T cells in the tumour, years before, 6 months, depends what the timing is, before you received immunotherapy, your response to immunotherapy is predicted by this. I wanted to get some new data in here so I could sort of show you guys not only published stuff, but the question we've asked is why do some patients have a cancer that is very immunogenic versus not? It seems to be this intrinsic feature that stays in the cancer for the full disease course of a patient. There must be some feature of the cancer that makes this happen. We all sort of know that tissue biopsy, RNA-seq and genomics is a bit challenging because you get all the cells, you don't quite know if you're getting RNA from a cancer cell or a fibroblast or an immune cell. So we've taken this approach here where we can isolate 100% pure cancer cells from a patient's tumour to sort of study what is the biology of that cancer related to immune infiltration or not. We've made a lot of surface markers where we can get down, perform flow cytometry, sort out cells. What I'm showing on the left is just healthy kidney. It's EpCAM very high. All the cancer cells have lost EpCAM. They've turned on these markers CD24, ENPP. And we can actually find 2 populations of cancer cells in most of our patients. When we sequence them, they're extremely pure. So you can see both of these cells have lost chromosome 14, or the part of the chromosome 14. There is a clonal relationship between these cells, so there's some development going on. You can see here, this is the chromosome 3 loss. The low population of cancer cells haven't really lost this part of the chromosome yet, but the high cells have lost 1 copy. Same with things like VHL mutations. You can see this, the left cell, the pink one, the 24 lows we call them, it has the VHL mutation, but then the 24 high cell has lost the good copy, so 100% of the reads are now VHL positive. And this tells us it's very pure cancer. 100% of the DNA reads we recover have the mutations present for the cancer. There's no fibroblasts or immune cells contaminating this analysis. So we did this for around 100 patients. We sorted these populations of cells. We did DNA and RNA sequencing to learn what's different about hot and cold kidney cancer cells and why do they respond to immunotherapy. The first thing that came out was all oxidative phosphorylation. All the patients that progressed later on immunotherapy and didn't have very good immune responses had very high levels of oxidative phosphorylation in their cancer cells. And if we correlate just the CD8 infiltration with this oxidative phosphorylation score, this was one of our strongest correlations. So that R squared is wrong. It should be around 0.4, minus 0.4. We can quantitate this in the cells, so because we can stain the pure cancer cells, we can now start to stain their mitochondria, mitochondrial potential, and all sorts of things like that. All the patients who progressed on immunotherapy later had very high levels of mitochondrial potential in their cancer cells, yeah. What was interesting to us, though, is we're trying to understand the biology of immune cells in the cancer, what are driving them, what are making them work. This is what I'm showing you here on the left is kind of a low OXPHOS patient. What you can see is we've gated onto the T cells. We have the stem cells on the left and these more effector-like cells on the right. This patient has everything going right. They've got a stem population. They're generating the cytotoxic cell. This is a good immune response. And we sort of know you need all that happening to respond to PD-1. On the left, in all our high OXPHOS patients, the stem cells seem to make it to the tumor. We find these little niches, but that last step of them generating the killers is for some reason broken. And we've done this in over 100 patients now. It's very significant, very interesting. And then this is the actual survival data in response to checkpoint therapy. So the patients that we originally sequenced that went on to checkpoint therapy, if they had this high OXPHOS score in their tumours, they were much more likely to progress. No immune response, no response to the therapy in the blood. So our conclusions from this. T-cell activation in the blood can tell you a lot about the response to immunotherapy on the first cycle of checkpoint therapy. There's a lot of information right up front, and it gets worse as time goes on. Having an immune response or not seems to be an intrinsic feature of the cancer. If the cancer was immunogenic at the time of surgery, it seems to later respond to immunotherapy later on. And then there's some sort of new data that we're generating. This oxidative phosphorylation status in the cancer cells is related to if you can actually generate an effector anti-cancer T-cell response or not, and this can be a strong predictor of response to immunotherapy. So I think understanding what's happening there is our current focus. Thanks to our lab, particularly our doctors, Viraj Master, who gives us lots of tissue, Asim Billen, who recruits tons of patients to trial, all the students who did this work. Thanks a lot.
Thank you. Our next speaker will be Marietta Thoma from the University Hospital in Bonn, who will speak about patient-derived organotypic cultures predicting immunotherapy responses.
So, hello everybody. Thank you very much to the organization committee for inviting me here. And I would like to talk about organoid cultures for predicting therapy response in clear cell renal cell carcinoma. And the learning objective, how to predict a therapy response using different types of organoid-derived cultures. Well, the renal cell carcinoma is the third type of urological cancer, and we have in Bonn about 75% of clear cell renal cell carcinoma and 25% non-clear cell renal cell carcinoma. And we know that clear cell renal cell carcinoma has a poor prognosis with roughly 30 till 50% of patients having relapsed metastatic disease at diagnosis or in follow-up. The therapy management is risk group driven, and we have favorable risk group and intermediate and poor risk group with several therapy combinations, and also second and third line therapies. The risk score assessment is made by IDMC, and you have roughly clinical risk factors, and you made 3 groups. And favorable group has no risk factor, the moderate 1 to 2 risk factors, poor more than 2 risk factors. And it's validated also in CCRCC, also in non-CCRCC. But like I said, it's clinical and it doesn't concern the tumor. And on the molecular side, we have also some prognostic factors, like a high angiogenic signature. And it seemed to be that those tumors with high angiogenic signature have a better outcome under anti-angiogenic therapy. They are tumors with high T effector signature. They have a better outcome under immune checkpoint inhibitors, and also the PD-L1 can be used as prognostic factor. It was reported to be positive in 23% of ccRCC and 10% of papillary renal cancer. And the PD-L1 positive tumors, they have longer progression-free survival under IO therapy, but in two studies, no differences in overall survival. So to me, I am a pathologist, and we are working with tissue and also with fresh tissue. We are biobanking, and we ask ourselves, can we do better? Can we have prediction of therapy response, an individual prediction of therapy response? And so we try to establish patient-derived organoid models. What's this? Organoids are derived from few cells of a tissue, or you can derive them from pluripotent stem cells or adult tissue stem cells. And they are simplified, but three-dimensional structures with realistic microanatomy. And the organoids have the ability to self-renew and differentiate. It's only a point. You have here only the epithelial compartment. And how to do that? You have your tumor, and you take a small piece of tumor. You are dissociating this. You have single cells, and these single cells can be taken in matrigel. You need a matrix in order to grow your organoids, and put it on a dish, and you adding some special media with a lot of growth factors. Don't forget FGF10, and you can grow organoids. And you see from bottom till here down how the organoids are growing. Here there are three patients. It's a work of Kazama et al., published 2021. And those organoids, they are round structures, and they are growing bigger in 4 weeks. And if you are taking a look at the histology, the organoids are quite similar to the tissue of origin, to the tumor of origin. They are having also clear cell and organizing their self as the tumor of origin. And what can you do with such organoids? They are patient-derived, patient-specific. You can analyze like tyrosine kinase inhibitors specific for each patient. And you can see, for example, axitinib, not all the patients are having the same answer to axitinib therapy. And that could help to choose the right tyrosine kinase inhibitor. Well, it's very nice, but you still miss some components, and you are missing the stromal component, you are missing the immune cells. And 2018, Neil et al. Published a paper in Cell. He's working, or was working, in the lab of Kelvin Kuo in Stanford about air-liquid interface organoids. And they are taking also, they took also from primary tumor a small piece, and you can mince that in even smaller pieces of tumor, put that on an insert with collagen, and you have on the top your air interface, and you are putting the insert in media. And what you need to have is also IL-2 to keep the T cells happy. And the origin histology is reproduced. The preservation of, you can preserve the complex tumor microenvironment, mid-stromal and immune cell infiltrate. T cell receptor repertoire was conserved, and you can reproduce the PD-1, PD-L1 checkpoint. We were very happy to read about it, and took a lot of tissue and tumors in culture, and a lot of kidney tumors. The most of them were ccRCC. And if you take a look on the organoids, they are more complex. They are not round. They have a complex architecture. And also here, the origin histology is reproduced. You have clear cell growing in this collagen matrix. And we made some immunohistochemistry because we wanted to know what is growing there. And you see on the left side, on the red arrows, they are the PEX8 positive tumor cells. Then we have a lot of CD8 positive immune cells inside, outside also, but inside the organoid we still have the CD8 positive immune cells. And we have also FAP positive stromal cells, fibroblast-associated protein positive. So we could preserve the microenvironment. Then we took a look how similar they are, the organoids to the primary cell culture. And you see they are clustering very nice in RNA-seq. And on the waterfall volcano plot, you see that there are some genes being differentiately expressed, about 400. This is not that much for culture. And you see that on the left side, we have a lot of downregulated genes, and they were mostly associated with hemoglobin. And our explanation was we don't have circulation. It's a complex structure, but without vessels. And that's why the hemoglobin is downregulated. And on the right side, we have the upregulated genes, and they are mostly involved in inflammatory response, IL-2, STAT5 signaling. And our explanation was we are giving IL-2 in media, and probably it's because of this additionally IL-2 in the media. So we wanted also to see, can we predict therapy response? And treat 10 organoids with cabozantinib and nivolumab for 1 week. And then after 1 week, we embed the organoids in paraffin and then quantified the necrotic areas. And you see on the left side the controls. They are quite viable and quite nice. And on the right side, the cabozantinib response, and the areas still viable, they are marked. And with nivolumab, we had in those 2 cases the best response. We did then enlarge the cohort, and now we have 23 early PDOs from CCRCC treated with nivolumab and cabozantinib, and 14 have a good response to nivolumab, 9 have no response. Cabozantinib, it was half-half, with no differences in PD-1, PD-L1 status. And we took also a look at the mutation status. There are some differences, but they are not statistically significant. And then we thought, well, what is the difference between cases which are answering to TKI and cases answering to immune checkpoint inhibitors? And those are those 2 groups. And we found also the differences not in the T cell signature, but in the B cell signature with the Ig immunoglobulin genes being differently expressed. And we were so happy to see this paper because it confirms what we are also seeing. And to wrap up, the take-home message, patient-derived organoids from CCRCC could be cultivated as in-dome culture or air-liquid interface format differently, and you can preserve the tumor histology, also the genetic aberrations, and allows us to screen for the best therapy combination and for discovery of new therapeutic options. Thank you very much for your attention. I want to thank the patients who gives us the informed consent to do this study, and to my group, and all our collaborations partners. Thank you very much.
Now it's my pleasure to introduce our co-chair, Annika Fendler, who will be speaking about transcriptomic heterogeneity in RCC. And I want to mention that it's very exciting that just a few months ago, Annika established her own laboratory at the Charité University Hospital in Berlin. So maybe you could take a moment later today to congratulate her on that. I'll turn it over to you.
Thank you very much for that nice introduction. I will talk about transcriptomic heterogeneity in CCRCC today. So my learning objectives are what actually is transcriptional intratumoral heterogeneity and how is it associated with genotype and clinical phenotypes in CCRCC, and why is it important to consider this heterogeneity when we're developing either prognostic or predictive biomarkers. So when we look at the tumor, and we saw a similar picture earlier in this session, there's a lot of heterogeneity in the tumor which we can all appreciate, and it comes at certain levels. One is the genetic heterogeneity, which kind of shows how the tumor evolved over time, but we also have different tumor microenvironment compositions, and we have different organizations spatially and crosstalk between cells, but also within the tumor cells, there is plasticity either in how the cells are differentiated or how the capacity to metastasize is, or what cellular programs are active. So transcriptional heterogeneity that we're seeing in bulk RNA sequencing can be controlled by all of these factors, either genetic, epigenetic, or non-genetic factors. So there have been a lot of single-cell sequencing studies, or several in the last years in kidney cancer, which really have given us a great overview about the composition, mainly of the TME, and what kind of cell types we're finding in different kidney cancers. And these are just two examples of the studies that have been published. The limitation maybe of single-cell sequencing is that we can't usually assess it in a large cohort of patients in multiple regions from the same tumors, and that this doesn't allow us maybe to assess systematically how transcriptomic heterogeneity, genetic heterogeneity, and clinical phenotypes are linked. So within the TRACER-X, the study I was working in together with Samra in the last years, we wanted to systematically assess transcriptional heterogeneity in clear cell renal cell cancer in a multi-regional data set and understand what factors drive transcriptional heterogeneity and how this transcriptional heterogeneity associates with clinical outcomes. So I just wanted to go back a little bit and talk a bit about TRACER-X and the genetic data. So as I said, it's a multi-regional sequencing study, as you can see on the left side. I think what's important is to see that we did an analysis where we wanted to see how many samples you need to capture genetic heterogeneity, and you can appreciate the more heterogeneous the tumor is, of course, the more biopsies you would need to appreciate all the changes you can find within the tumor. And then if you look at whether this is even important, I think it is. You see that intratumoral heterogeneity on the genetic level is associated with outcome, with progression-free survival in kidney cancer. And something else I want you to appreciate is that also the chromosomal complexity of these tumors is associated with outcomes. So this really led to this model where how the tumor evolves is associated with a clinical outcome and the proportion of tumors that will relapse and how fast and metastasis will grow. And this is associated with the chromosomal complexity and the heterogeneity of the tumors. So we took regions from this cohort and performed bulk RNA sequencing, and we did this in a total of 232 regions of 80 of these tumors. The median of what we sequenced was 3 regions per case, ranging from 1 to 13. And we selected cases so that we had a good representation of genetic alterations. You can see that on the right-hand side. And we also sequenced, if available, a metastasis from the same patient and the primary region in the tumor from where the metastasis likely arose. So if we look what can drive intratumoral heterogeneity in these tumors, then interestingly, genetic intratumoral heterogeneity was not what was most closely associated. So this is the proportion of subclonal and clonal driver mutations. But what was maybe not surprisingly really driving is the tumor microenvironment, and also to some extent chromosomal complexity, which maybe also might not be be explainable because you get large aberrations and changes in the chromosome. We also then did a multivariate analysis to really see how this plays out when we take several genetic and clinical factors into account. And again, here we confirmed that chromosomal complexity was quite important in driving transcriptional heterogeneity. So again, the question is then, is this just like an artifact and it doesn't really matter? But we think it does matter because also, just like with genetic heterogeneity, transcriptional heterogeneity in itself is prognostic. And this is interesting because it seems to cover some additional information because it's not associated per se with genetic intratumoral heterogeneity. And why is this important? So we looked at certain biomarker sets that have been described in the kidney in recent years. We looked at prognostic biomarkers and predictive biomarkers. And then we checked for each region, how would we classify the tumor based on this signature. And on the right-hand side of each of these graphs, you can see per column a single patient, and you can see the dots as every region. And you can see there's quite a big proportion of tumors where we would misclassify, where one would say the signature is present and the other region would say it's not. And you can see across all these different data sets, it's around 40 to 50% of the patients. So this really makes you cautious when you're designing biomarkers. You have to account for this heterogeneity because when you take a single biopsy and would make a treatment decision based on that, you might misclassify the tumor because you're missing something within the tumor. Sorry, it's not going on. Yeah, maybe I just continue. Maybe the slides, no. Okay, so, and we were not the only ones who are finding this. I can't show you now, but Ari Hakimi's group has published a paper last year where they also looked at multi-regional RNA-seq data, and they found for the MOTSA molecular subclassification that they see a similar effect that they could misclassify by just a single biopsy. So to conclude, we see that there is genetic and transcriptomic heterogeneity and that these are pervasive features in CCRCC and are associated with different clinical behavior, and that transcriptomic heterogeneity is not only dependent on the genetic heterogeneity but on a multitude of other factors, the TME and also the chromosomal complexity. And we see that it's very important to consider transcriptomic heterogeneity and sampling bias when we're developing transcriptional biomarkers and thinking about introducing them into clinical trials, because we have to minimize the risk of misclassification. Thank you.
Thank you very much, Annika. The final speaker in this session is Jan Alexander Vanno from the Georges Pompidou Hospital in Paris, and his topic is optimizing clinical trial design by integrating tissue and tissue-derived markers. Thank you very much.
Thank you very much. Dear colleagues, good afternoon. I would like to thank the organizing committee for giving me the opportunity to present this important topic. This is optimizing clinical trial design by integrating tissue and derived markers. The learning objectives are the following: to remember the unmet medical needs in the management of metastatic RCC, to think about the biomarkers that might be ready for clinical use. I will not be too much exhaustive because we talked a lot about this topic this morning. To know the ongoing biomarker-based trials. There are few biomarker-based trials, but I will not be exhaustive too. And to identify the challenges of integrating biomarkers into the design of clinical trials. Why do we need to optimize clinical trial design? This is a very simple question, and we have simple answers. First, we have too many options, and we saw it a lot of time this morning. We have 3 TKI-IO combination for nearly all metastatic patients in the first-line setting. For 3/4 of them, we have a 4th option with Nivo-IP for intermediate and pro-risk group patients. And we have 3 to 4 options. We have one choose to make in the first-line setting, and it will impact the efficacy of second-line setting as we saw this morning. So we have probably too many options, and we have just to choose one option, and we don't have to miss the point. And then we have also other concern that efficacy can always be improved. I take this example from the Checkmate 214. 40% of patients died or are progressing in the first 6 months of therapy. We do not want to do that with our patient, but at the opposite, we have 30% of patients that are still not progressive at 5 years, at 60 months, which is very impressive, and this is what we want for our patients. But combining all drugs is not obviously the solution, and I take this picture from the Checkmate 313. You know that this trial is positive on progression-free survival in intermediate poor-risk patients. The triplet Cabo-Nivo-EP is superior to Nivo-EP, but if you look at the poor IMDC risk group, the triplet is not superior in terms of progression-free survival over Nivo-EP. So maybe combining all drugs all together without selection is not a real solution. What could we use? We talked a lot about biomarkers this morning. Just very simple biomarkers. I think we have to reconsider PD-L1 as an interesting one biomarker because it is quite simple to do. And if you look at the efficacy of Nivo-EP regarding progression-free survival according to the PD-L1 status, obviously you see a huge benefit of Nivo-EP in patients with PD-L1 positive tumors. Whereas with TKI-IO, as an example, I put it the Nivo-Cabo Kaplan-Meier curves, you see no difference in progression-free survival according to PD-L1 status with the same cutoff and the same clone. Maybe we have a space here to do something, and I will show you some initiative later. The sarcomatoid component probably may matter a lot. You know that is a poor prognosis factor. You see on the right the median progression-free survival with sunitinib in case of sarcomatoid component. This is very poor PFS rate. But look at the data with Nivo-EP, really impressive data. 23% of complete response, 26 months of median PFS, which is really impressive in this poor prognosis population. But the story is not the same with Cabo-Nivo and Lenva-Pembro with TKI-IO, because in case of sarcomatoid component, look at the median PFS, there seems to be lower in case of the sarcomatoid component than in case without sarcomatoid component. And probably there is another space here to put it into new clinical trials. We talked this morning about gene expression signature, and we have to congratulate the big efforts that has been done on the immersion trial where with David McDermott, who described the androgenic anti-effector gene expression signature. And of course, these gene expression signature are not often have not been validated on Checkmate 214, but it seems to be still very interesting to integrate it into biomarkers. And in the same trial, Bob Moser and colleagues have a different approach by an unsupervised clustering, and they describe 7 distinct clusters with different RNA profiles, distinct DNA alteration, and distinct biological pathway. And some of these pathways and these clusters were associated with atezo-bev or sunitinib superior efficacy. I think it's very important to see this. How could we translate this data into clinical trial? And I will show you later a new initiative on that topic. And just to remind that 8 years ago, we already described a 4-molecular group by unsupervised clustering in 100 patients treated with sunitinib. And we found 4 different molecular groups were associated with distinct biological profile, distinct TME infiltration, and distinct sensitivity and resistance to sunitinib, with CCRC1 and CCRC4 being the worst prognosis. And based on this preliminary data, we launched the Bionic trial that we already talked this morning, where we randomized patients in first-line metastatic setting between Nivo, Nivo-EP, and VEGFR TKI according to the molecular group. Patients in group 1 and 4 were randomized between both IO arms, and group 2 and 3 were randomized between TKI and nivo-EP. I will not go through all the results of the bionic trials. We reported them last year, and we have updated it at the last ASCO GU, but just to say that it is feasible to integrate gene expression biomarker in a clinical trial. And thanks to all the collaborators, Bionic continued to generate key data. Florent Petit-Prétol's about tertiary lymphoid structure. Maxime Mélan presented this data last year at the last ESMO. I presented the updated data at the GU ASCO. And we have a very fruitful collaboration with Gabriel Malouf on the methylation profile and the fibroblast. So we have other biomarkers. Bionic is not the only biomarkers. We have this very great initiative from Brian Rini based on the 7 clusters that I already mentioned on the work from the work of Bob Motzer. And patients will be will not be randomized, will be treated according to the gene expression profile. Roughly, cluster 1 and 2 are clusters, pro-angiogenic clusters, and this patient will receive the nivo-cabo. You can see the ambitious primary endpoint of objective response rate, more than 75%. And cluster 4 and 5, let's say inflammation and proliferation profile, this patient will be treated by nivo-EP with, again, a very ambitious overall response rate. Note that 3 clusters must be excluded from the study. It's about 1/3 of the study, probably because they don't find any good space, good treatment for this patient. But we have to follow this trial, and I think it is currently recruiting. And we saw a very pragmatic and important trial launched by Laurence Albiges. We will be recruited soon. It's a CARE1 trial. It's a randomized phase 3 trial based on PDL1. So I mentioned that PDL1 is probably a very useful target, and the trial will compare double IO and IO-TKI according to the PDL1 status. The hypothesis is that in PDL1 positive tumors, the double IO is superior to TKI IO, and in PDL1 negative tumors, the double TKI IO-TKI is superior to the double immunotherapy. And 102,000 patients are awaited in this trial. How to, just one slide to say the several issues that we have to address. First, of course, the biomarker is at the center of the topic. It would be predictive more than prognostic. It should be reproducible, of course, but we have to think about the tissue. Is it blood or tumor tissue? If it's tumor tissue, we are just talking about the tumor heterogeneity, which is very important between primary versus metastasis. Is it FFPE samples that we have to use? Is it frozen? In Bionic, we use frozen. We know that it's difficult to use frozen as a routine, as a clinical care. We have to think about the procedures, which technology will be used, RNA sequencing, a gene panel, a limited gene panel. What is the cost per samples? Do you need a centralized analysis, or could you do it routinely in the local lab? And the results, we have to anticipate the failure rate, because when you include patients, you have to know how much do we miss to identify the right biomarkers. And of course, I think it's the most important here is the delay to obtain the result, and the delay should be compatible with care. And the Bionic trial, we have the delay of 2 weeks between the sample reception to treatment molecular grouping. So the take-home message are very simple. We clearly need to optimize the clinical trial design. We have already promising biomarkers available and almost ready to use. We have several issues that must be addressed at the very early step of the trial concept design. And we have already exciting, but too few, but exciting biomarker-based trial underway, and let's do it. And we have little to lose, and our patients have much to gain. Thank you very much.
We have a few minutes for questions. If people could please come to the microphone and identify yourself, that would be great.
congratulations, fantastic work, all of you. My question is with regard to intratumoral heterogeneity. So my question for you is, at the end of the day, you feel we should only look at the transcriptomic heterogeneity, or we should have some kind of bulk assessment of the intratumoral heterogeneity to use that as a surrogate, as a biomarker in itself?
I would argue that looking at it as a biomarker itself is helpful, but obviously also complex to achieve because it's a compound score which has to take into account a variety of regions from the same tumor. I think what's important is understanding this also more and then trying to find, if we look at transcriptomic biomarkers, and it might not be the only thing we want to look at, are there markers which are more stable, which even though we only look at a small part of the tumor can still give us the important information on, for example, when to treat or not to treat.
And so what's not clear to me is how do you assess it? What would be the right way to assess this intratumoral heterogeneity that could be applicable?
So obviously we can do it in a multi-region cohort, but that's not clinically feasible, right? The other thing is to look at something that is stable across an individual tumor, but not across tumors from different individuals. And then the other thing, something that also Samra was leading on, is she is taking leftover tissue and then looking at the compound of this. Or looking in the blood, for example. I think it's still important. I mean, there's issues with looking at markers in the blood, but I think Hayden's work is really nice, seeing this boost of activated T cells that you can see, and then this is associated with the response.
Yes. Sorry.
Okay. I think I was just a little heavy, but hiding in the back. So Hayden, that was a really nice talk. I was just wondering, it seems like the data you showed was all with Nivo-IPI. Have you looked at whether you see those T-cell bursts with just PD-1 monotherapy or any of these combination TKI-IOs?
Yeah, it should have been a bit more clear. So that cohort was a combination of PD-1 alone, PD-1 IPI, and then there's also some of the Nectar-IL, a very small number of the Nectar-IL2 plus PD-1. So it was a mixed group. What's really interesting is it didn't matter. There was no difference between the different groups or anything like that. And then just to add to that, so we just did this recently on, I think it was the neoadjuvant Exelixis cabozantinib trial. We also see a burst in the activation on that as well. And we haven't done a correlation with survival yet. We're sort of still waiting to see, but it seems to be sort of, maybe more than just immuno generally.
And just very quickly, do you think that seeing that burst in the first dose, or maybe maximum at most by the second dose, means that that's all you need to activate the immune system, and then you need to just maintain it after that?
Yeah, I do wonder about dosing and timing, and also, the 3 or 4 week is a bit of a guess that we sort of started with and now we're stuck with. But, maybe if we can look over longer time to see if it drops off and can be reboosted later and things like that. I think there's a lot to learn on those as we go.
Agreed.
Thank you.
Thanks.
Should we come back to the front microphone?
Yes, good afternoon. Yes, I would like to, I have 2 questions actually. The first one is, do you know exactly where the activation of CD8 T cells response is going from lymph node to blood? That's my first question. And my second question is lymphadenectomy not harmful and in charge to treat the cancer?
Yeah, so our recent paper that came out a couple of months ago was all about the T-cell activation process in cancer. And so it starts in the lymph node, and they do this sort of weird thing where they become this stem cell and then migrate as a stem to the tumor, where there's a second phase of the activation where they activate. The PD-L1 works at all sorts of different levels of this thing. So in the lymph node, it helps the stems expand better and then migrate to the tumor. Then in the tumor, it helps them differentiate to the effector state better, and also help kill the tumor better. So that we kind of think that's why PD-L1 is such an effective drug. It's working at all these phases. Some of the other checkpoints are maybe working at 1 or 2, and things like that. What was your second part of the question?
Lymphadenectomy. Do you think it's harmful or helpful?
I have an older immunology colleague who said, if I have cancer, I would like to keep my lymph nodes. So I think, that's right. I think that the lymph node is very important. those stem cells, there's about 10 to 100-fold more of them in a lymph node than in the tumor. So a lot of your immune response is actually held back. And so I do think it's an important, how to mobilize them and use them is important.
I think we'll go to the back microphone, and both people in the back, I think, have been waiting the longest, so we'll take those two next.
Rachel Giles, International Kidney Cancer Coalition. So firstly, to the whole panel, excellent topic. I think this is probably the biggest unmet need from the patient perspective, is this need for a development in these areas. And so we're very happy to see this. And I think I speak for Gretchen and Dina as well in this room as far as the patient perspective. But there's also a bit of a frustration. When we see our colleagues in lung cancer, breast cancer, even melanoma, they have this figured out, and they have this functional in the lab, and it's something that's directing their treatment. And the comments that Jan made at the very end sort of raised the question is, why can't we just figure this out just like they have?
Anybody have an answer?
It's a bit of a tough question, I know, but there's a lot of frustration from the patient community that this has not been addressed it's been more difficult than we thought, and it's not been addressed as thoroughly as in other cancers. So if someone could comment to that.
I agree, totally. I think we need combined efforts in figuring this out, like really working together. And I think the last slide was actually great, saying we have to integrate these also into clinical trials, and we have to do it because we have nothing to lose, but there needs to be also more fundamental understanding of biology to then also know. So it's a combination. But
I understand entirely. I'm just pointing out that the research from the patient perspectives have clearly shown that this is the #1 priority for advancing care at this time. So thank you so much for this important research.
Thank you. We really appreciate hearing the advocate position. Thank you very much. Maybe we'll take the question in the back, then we're very short on time, so if you have a short question, that would be ideal.
Sure. Just a quick comment on the last thing. We had a little brainstorm last night with the French colleagues about differences between melanoma and kidney cancer, and the conclusion was that kidney cancer is more complicated, so maybe that's part of the answer. It's a quick question for Florent, and beautiful work, everyone, but just I'm interested in the temporal dynamics of TLS formation. I mean, are they being formed all the time, and how is that relevant to response?
So it's still a bit unclear. We don't really know what causes them to appear in some tumors, and it's only in a fraction of tumors that they do appear. What starts now to be quite known is that they arrive sometimes with different degrees of maturation. It would seem that it's actually the most mature TLS, so those that have a fully formed germinal center that would be the most efficient to promote survival and response to immunotherapy. But it's still unclear what causes the TLS to appear.
Mm-hmm. Thank you.
We have two more questions. So first,
we have start.
I have just a question about oxidative phosphorylation, because you showed that it's associated with resistance to IO. So when we lose VHL, usually we activate hypoxia and we activate glycolysis. Can you please explain to us, because VHL is lost in the majority of clear cell RCC, what's the balance between oxidative phosphorylation and glycolysis? Yeah. How it's shifting? Do we have the same in the same tumors? So I'd like to have your clarification.
This data is a month or two old, so we're still thinking about it. There's no correlation between the glycolysis and oxidative phosphorylation, which was really interesting. Seems like they're not switching, they're doing each independently, which was interesting. No correlation with the oxidative phosphorylation and hypoxia, a little bit with the glycolysis and hypoxia correlations. So I think we just have a lot to learn about these systems. People sort of say that they're a trade-off, but I don't think they are in the cancer cells. They've found a way to do both. They're using them both for energy. And then after treatment, we have no idea. We don't know how they play into it or anything like that. We're looking at that in some xenograft models now to see how they change, but lots to learn, sorry.
Yeah, one quick question. So congratulations to all for those very interesting presentation. I have a question for you about the blood evaluation, looking at the stimulation of the immune system. You showed that if you don't have a stimulation that's cycle 1, you may have no further activation and the patient may progress. So could you say that recommend that maybe if you don't have this kind of overexpression or stimulation, well, we can maybe quit. At the opposite, if you have a good stimulation, can we reduce the cycle of ipilimumab, from 4 to 3 and to 2? Michael Atkin asked a question just before, but what is your response to that?
Well, my response is we should do a trial to see if that's the correct answer. But that is my hypothesis, that we should be able to know on the first cycle if we should keep going or not. And I think it plays into some of these neoadjuvant sort of things and adjuvant therapies. Maybe if we know on the first cycle, we stop doing it. And, but then the other part of that is we don't have a lot of other options if you fail PD-1, so because everything's combination now. So it's a little hard to know what the decision is to make. We're talking about this now to do it prospectively and work it out, but that is the hypothesis.
I want to just thank all of the speakers for a really wonderful session that obviously generated a huge amount of interest and discussion. And stay put, because the next session, session 11, will start momentarily. Thanks, everyone.
Thank you.